You know, the artificial intelligence that can generate an image from a text prompt or power a chatbot conversation is now being applied to one of the most fundamental questions in science: understanding the building blocks of life. We are currently witnessing a significant shift in biology, a move away from the slow process of observing nature toward rapidly predicting its mechanisms, and now, even to the point of creating entirely new molecular structures.
So how exactly did this happen? Well, throughout this explainer, we'll first connect the AI that many of us are familiar with to the world of molecular biology. Then, we'll explore the new toolkit scientists are using, see how it addressed the long-standing protein folding problem, and look at the new frontier of designing novel proteins. And to wrap up, we'll consider what these developments mean for the future of science.
Let's begin with the artificial intelligence that has become a fairly common part of our digital lives. Many of us have seen AI that can understand and generate human language, like ChatGPT, or models like DALL-E 2 that produce complex images from a simple description. We also see computer vision at work in autonomous vehicles, helping them interpret the visual world. The common thread here is that all these systems are designed to find patterns. And as it turns out, the biological world is dense with intricate patterns. So, what is the link between these systems and molecules? It really comes down to a change in perspective. The key insight was the realization that biological challenges could be reframed as the kinds of problems these AI tools were already designed to solve: problems of language, vision, or network analysis. This brings us to a new toolkit for biology. It functions by viewing proteins from several different angles, and each one of these viewpoints allows for the application of a specific type of deep learning model. You see, a protein's properties can be represented in multiple ways. We can treat its linear sequence of amino acids as a line of text. The proximity of its components in 3D space can be mapped out as an image. The web of interactions between its atoms can be modeled as a network. And the very process of it folding into its final shape can be seen as analogous to an image generator creating a clear picture from a random starting point. So let's look at the first one: large language models, or LLMs. These models are trained to understand context and relationships within sequences, like words in a sentence. By treating a protein's sequence of amino acids as a sentence written with an alphabet of 20 letters, LLMs can learn the grammar of protein structure, discerning which amino acids are likely to be found near one another. Next are convolutional neural networks, or CNNs, which are central to computer vision. They are very good at identifying patterns in images. A protein contact map, which is essentially a 2D chart showing which parts of a protein are close to each other, can be processed like an image. This allows a CNN to detect recurring structural motifs, the common shapes that form the building blocks of a protein's final structure. Then we have graph neural networks. These are built to analyze network data. A protein can be modeled as a complex network, where each amino acid is a node, and the forces between them are the connections. GNNs can then analyze this network to understand how different parts of the protein, even those far apart in the sequence, communicate and influence one another. And finally, there are denoising diffusion models. These are the models behind many of the popular AI image generators. They work by starting with random noise and progressively refining it into a detailed image. This process serves as a compelling analogy for how a long, flexible chain of amino acids collapses from a disordered state into a single, highly specific, three-dimensional structure. All right, so with this new AI toolkit available, we can now turn to how it was applied to one of the most persistent challenges in biology: the protein folding problem. A protein's function is determined almost entirely by its 3D shape, but determining that shape through laboratory experiments is a very slow and costly process. For about 50 years, the goal of simply predicting that shape from its sequence of amino acids was a major unsolved problem. The sheer number of possible ways a protein chain could theoretically fold is astronomically large, making it a formidable computational challenge. Progress toward a solution was slow, and then it became very rapid. For a long time, the most effective methods depended on having a known structure from a similar protein to use as a template. The first AlphaFold system in 2018 showed significant progress using CNNs, but it was AlphaFold 2 in 2021 that represented a genuine breakthrough. It used a completely new architecture that effectively solved the prediction problem for a large number of proteins. And now, AlphaFold 3 is expanding these capabilities even further. So, how does a system like AlphaFold 2 work?
It can be thought of in two main phases. First, a component called the evoformer analyzes the protein sequence alongside sequences from its evolutionary relatives. It gathers clues about which amino acids are likely to be in contact. It then uses an attention mechanism to cross-reference these clues and build an abstract representation of the protein's geometry. Then, a second component, the structure module, translates this abstract information into a final, detailed 3D model of the protein's atoms. The effect of this development has been substantial. This approach has now been used to predict the structures for more than 200 million proteins, which covers nearly all cataloged proteins from organisms across the tree of life. To put that number into context, let's consider the Protein Data Bank, or PDB. This is the global archive for all experimentally solved structures. After more than 5 decades of laboratory work, it contains around 220,000 structures. In just a few years, AI-based prediction has expanded this body of structural knowledge by a factor of nearly 1,000. The field continues to move forward. The next generation of models, including ESM-fold and the latest version of AlphaFold, are now moving beyond single, isolated proteins. ESM-fold, for instance, is a large language model that can generate a structure prediction from only a single protein sequence, without requiring data from evolutionary relatives. AlphaFold 3 can now predict the structures of molecular complexes, proteins as they interact with DNA, RNA, or small molecules like drugs. This is important for understanding how most biological processes function, and its structure module now uses a diffusion process, which links back to the AI image generation methods we discussed earlier. Now, having developed the ability to accurately predict a protein's shape, the scientific community is turning to a new objective, not just interpreting nature's existing designs, but creating new ones. We can think about structure prediction and protein design as two sides of the same problem. Prediction takes a sequence as input and asks, what structure does this form? The reverse of that, known as inverse folding, starts with a desired structure and asks, what sequence is needed to form it? Solving this inverse folding problem is a critical step toward computational protein design. The ability to specify a target shape and then find a sequence that will adopt it opens up the possibility of engineering proteins for specific functions. And again, researchers are applying the AI toolkit to this challenge. Some models approach it as a language translation problem, translating the language of 3D coordinates into the language of amino acids. Others use diffusion models to generate a sequence that is conditioned on a given structural backbone. A third approach involves running a prediction network in reverse, searching for a sequence that maximizes the network's confidence that it will fold into the target shape. However, the ultimate goal of the field is de novo design, which means from the new. This involves designing a protein with no known precedent in nature, creating both a novel structure and the specific sequence that will produce it. A current workflow for this, used in a tool called RF diffusion, combines several of these AI techniques. The process begins with a random cloud of atoms, essentially digital noise. A diffusion model then denoises this into a coherent and physically plausible protein backbone. Finally, an inverse folding tool is used to determine an amino acid sequence that is likely to fold into that newly designed shape. These advances in both prediction and design are not just theoretical exercises. They are beginning a new era in biology, with significant implications for both research and medicine. In experimental biology, AI is accelerating structure determination by helping to fit atomic models into data from electron microscopes. In drug discovery, similar generative techniques are being explored to design novel small molecules intended to bind to specific proteins involved in disease. And in biotechnology, the prospect of engineering custom enzymes for industrial processes or new therapeutic proteins is becoming increasingly tangible. These new tools are part of a general trend in AI, where models are becoming larger and more complex. The number of parameters, a rough measure of a model's capacity, has increased significantly. While protein-specific models are quite large, they exist in a landscape that includes general-purpose models that are even larger. This trend toward increasing scale is enabling new capabilities, and community efforts are working to make these tools broadly accessible. This period of rapid advancement has prompted reflection within the scientific community. As noted in the source material, there is a call to view this transformation not as a disruption, but as an opportunity to address scientific problems that were previously out of reach. This shift from prediction to design signifies a fundamental change in our relationship with the molecular world. We have moved from a primary focus on reading the existing blueprints of life to now beginning to write new ones.
the central question for the future is what we will choose to build with this new capability.